Hello everyone. In this video today we will be discussing about the hemostasis. What is hemostasis and what are the steps involved in the hemostasis? What exactly you mean by the term hemostasis? Hemo means blood and stasis means stop. So the hemostasis is the process where the leakage of the blood from the site of the vascular injury will be stopped. So this process of stopping the blood loss at the site of vascular injury, it involves platelets, clotting factors and the endothelial cells. So altogether all these factors, they play a role and they prevent the loss of the blood from the site of the vascular injury. Now, what are the factors which maintain the normal circulation? So, normally in the blood circulation, we don't have the clot formation or the stoppage of the flowing of the blood. So, what are the factors which help to maintain the blood in the fluid state? So, these factors, the factors which are mainly produced by the endothelium, we have a list of them. They are the nitric oxide, prostacyclin, adenosine diphosphatase, thrombomodulin, heparin sulfate, and tissue factor pathway inhibitor. We will see now how all these factors act, but just to remember them easily, a small wording NP, that if you remember these words, you can remember all the factors. N stands for nitric oxide, P for prostacyclin, T for thrombomodulin, H is for heparin sulfate, A is for adenosine diphosphatase, and T is for tissue factor pathway inhibitor. So these are all the factors which are produced by the endothelium. Now, when we see these factors, nitric oxide, which is produced by the endothelial cells, actually it causes the vasodilatation, that is relaxation of the smooth muscle and vasodilatation. Prostacyclin, it prevents the platelet activation and aggregation. ADPase, it actually breaks down the ADP which is released by the platelets. Now, ADP is one of the factor which causes the platelet activation and aggregation. So, together, nitric oxide, prostacyclin, and ADPase, they prevent the platelet activation and binding of the platelets to the endothelium. Now, the next factor is tissue factor pathway inhibitor. This is also produced by the endothelium. It's expressed on the surface, and this factor, tissue factor pathway inhibitor, it inactivates the tissue factor and the 7A complex. If you remember, tissue factor, it initiates the clotting mechanism. So this tissue factor pathway inhibitor, it inactivates this tissue factor 7A complex. And another molecule which is expressed on the endothelium is thrombomodulin. Now this thrombomodulin, it combines with the thrombin. When this complex is formed, it converts the inactive form of the protein C into activated protein C. And this activated protein C along with the protein S, it inactivates the factor 5 and 8. Usually all these clotting factors, they are produced by the liver and released in the circulation. So all these factors should be present in the inactive form so that the clotting doesn't occur. So these endothelial cells, they will be producing certain substances or expressing certain molecules which will be inactivating these clotting factors. So protein C, it will inactivate factor 5 and factor 8 along with the protein S. Then we have another molecule which is similar to the heparin, that is heparin sulfate, which is expressed on the surface. This heparin sulfate, it combines with antithrombin 3. And when it combines with antithrombin 3, this complex, it degrades the clotting factors, mainly thrombin, that is factor 2, then factor 9, factor 10, which are present in the circulation. So these are the mechanisms by which the clotting factors are inactivated. The clotting factors which are produced by the liver are inactivated in the circulation. Now another factor which is produced is tissue plasminogen activator. Now this activates the plasmin which causes the fibrinolysis. So if at all there is a clot formation, immediately it will be degraded by the tissue plasminogen activator which activates the plasmin and it will cause the fibrinolysis. So all these are the factors which are produced by the endothelial cells. I think now you remember the short form what I told you, that is NP, that is N stands for nitric oxide, P for prostacyclin, T for tissue factor pathway inhibitor, and H for the heparin sulfate, T for thrombomodulin, and tissue plasminogen activator, is it also stands for the T. Now, what are the steps in the hemostasis, or what are the components of the hemostasis? So, initially we have the vascular spasm. Immediately, whenever there is a damage, there is a vascular spasm. Then, after that, you have a platelet plug formation. So, till here, the spasm, vascular spasm, and platelet plug formation, we call it as a primary hemostasis. Later on, after the platelet plug formation, you have the activation of the coagulation factors. Then you have the blood clot formation. Blood clot means nothing but along with the platelets, you have other formed elements also, like the leukocytes and RBCs, all will be present in that clot. So we have a blood clot formation. Then we have a clot retraction and the repair. So till here, from the activation of the coagulation factors till the clot retraction and the repair, it is called secondary hemostasis. So primary hemostasis is just up to the platelet plug formation. Later on, formation of a complete blood clot and the clot retraction, it comes under secondary hemostasis. Now once the clot is formed, then again it undergoes the degradation by the process of fibrinolysis. Now we'll see each step, how does it occur. Now when this is a blood vessel, this is the endothelial cells, and you have the smooth muscle. So whenever there is a damage to the vessel wall, you have the loss of the endothelium which is covering this wall, and you have the first thing what happens is vasospasm. That is the contraction of the smooth muscle. Now why this contraction occurs is the endothelial cells which are present, they produce endothelin factor. This has a receptor in the smooth muscle. So endothelin immediately it acts on the adjacent smooth muscles and causes the contraction of the smooth muscle. Not only that, we have another mechanism called as myogenic mechanism. Means direct injury to the muscle causes muscle contraction. That is myogenic mechanism. And the third mechanism is the inflammatory mediators. They are released from the damaged tissues, and these inflammatory mediators, they stimulate the nociceptors which are present on the smooth muscle, leading to the smooth muscle contraction. So now you understood the vasospasm is caused by the endothelin, or it can be because of the direct muscle contraction because of the damage, or it can be because of the inflammatory mediators which act on the nociceptors which are present on the smooth muscle, leading to the smooth muscle contraction. Now what happens after this endothelium is lost from the surface? When endothelium is lost, the subendothelial collagen is exposed. Along with that, the von Willebrand factor is also exposed. This von Willebrand factor is produced by the endothelial cells and also by the subendothelial cells which are present. So this von Willebrand factors and the collagen is exposed to these two, von Willebrand factor and collagen. Platelets come and adhere. Now once these platelets adhere to these von Willebrand factors, they get activated. So normally the platelets, the structure of the platelets, they have alpha granules, dense granules, and thromboxane. So these alpha granules, they have adhesion molecules p-selectin, and they have the proteins involved in the coagulation such as fibrinogen, factor 5, von Willebrand's factor, and certain other factors which are involved in the wound healing like fibronectin, platelet factor 4, platelet derived growth factor, and TGF beta. These are present in the alpha granules. Whereas in the dense granules we have ADP. Then this ADP actually, when it is released, it binds to the receptor on the platelets and activates them. So we have ADP in them, ATP in them, ionized calcium, then we have serotonin and epinephrine. So all these factors, they are present in the dense granules. And thromboxane A2, that is present in the cytoplasm, this causes the platelet aggregation and the smooth muscle contraction. Now once there is a damage to the blood vessel and we have a loss of the endothelium, as I told you, there is the expression of the von Willebrand's factor and also the collagen which is present in the subendothelial tissue. So to this von Willebrand's factor and the collagen, the platelets adhere, and how they adhere is they have The molecules on the surface, glycoprotein molecules, mainly the glycoprotein molecule 1b, it attaches to the von Willebrand's factor, and another glycoprotein molecule, that is GP1A2A, it attaches to the collagen which is present in the subendothelial tissue. Now once the platelets, they attach to this von Willebrand's factor or the collagen, they get activated. Means normally the platelets have the smooth surface, they are round or the ovoid in shape, and once they get activated, they develop the spikes on the surface. Now this development of the spikes, it increases the surface area of the platelets. Now when they develop the spikes, they appear like a sea urchin. Now when this activation occurs, even the dense granules and the alpha granules which are present in the platelets, they are released. So secretions from the granules are also released. Now once the platelets, they get activated, there will be some conformational change on the surface also. So normally the platelets will have GP2B 3A molecule on the surface, but when the conformational change occurs due to the activation of the platelets, their affinity for the fibrinogen will increase. So this GP2B 3A molecules, their affinity for the fibrinogen will increase. So the fibrinogen attaches to these molecules on the activated platelets. And another important thing is there will be translocation of negatively charged phospholipids, mainly the phosphatidylserine molecule on the surface. So these two changes occur when the platelets get activated. Now, as I told you, there will be the translocation of the phosphatidylserine molecule on the surface. Now, once this phosphatidylserine molecule is expressed on the surface, there will be activation of the factor 12. The factor 12, when it comes in contact with this phosphatidylserine, it will be converted into factor 12A, activated factor 12A. Now what happens is when this factor 12A is formed, it initiates the coagulation pathway or coagulation cascade. How it is, we'll see. As I already told you that the expression of phosphatidylserine on the activated platelet surface, it will activate the factor 12. So this factor 12 will be converted into an active form, and this activated factor 12 will act on the factor 11. Now this factor 11 will be converted into activated form factor 11A, and this activated factor 11A will act on the factor 9, and then it will convert the 9 into active form, and this active factor 9 will combine with activated factor 8, and this complex, it will act on the factor 10 along with platelet factor 3 and calcium. So here this factor 10 will be converted into activated form, and activated 10A along with the factor 5A and along with the platelet factor 3, it will act on the prothrombin, converting it into the thrombin. Now this entire thing is intrinsic pathway because all these factors are present within the circulation, but they are present in inactive form. So they are activated, and then we have the end product as thrombin. So hence this one is called as intrinsic pathway, and it takes about 4 to 6 minutes to complete. Another pathway what we have to know is about the extrinsic pathway. Now here the pathway is coagulation pathway. It starts with the production of the tissue factor. Now this tissue factor, it will act upon the factor 7, converting it into its active form. Now this activated factor 7, either it can directly act on inactive form of the factor 10, converting it into the active 10A, or it can act on the 9, converting it into the 9A. So this we know 9A along with the factor 8A, again it will act on the factor 10, converting it into its active form. And once we get the activated 10A, it will combine with the factor 5A along with the platelet factor 3, and it will act on the prothrombin, converting it into the thrombin. So whether it's intrinsic pathway or extrinsic pathway, the pathway after the activation of the 10A will be similar. So this is a common point for the intrinsic and the extrinsic pathway. Now extrinsic pathway, it takes about 30 seconds. So this is a faster one, It takes about 30 seconds for the formation of the thrombin. Now, just the easy method how to remember. So, in the intrinsic pathway, we have factor 12, factor 11, 9, and 8. Now, these are activated in a sequence. Later, we have the activation of the factor 10. Then, from the factor 10, we have the activation of, along with the 5A, we have the formation of the thrombin. Then, from the thrombin, we have the formation of the fibrinogen. So, and in the extrinsic pathway, we have the activation of factor 3, the tissue factor, which will act on the factor 7, converting it into the active form. And this 7A will again act on the factor 10. So, 10 is the common point. Just to remember easily, the intrinsic pathway, you have the factors in the descending order. You have only the missing factor is factor 10 because this is a common pathway. So you have a descending order of the frequency 12, 11, 9 and 8. Whereas in the extrinsic pathway, that is which starts with the tissue factor 3. Tissue factor is factor 3. So to this we have to add 7 to get the 10. So along with the tissue factor, if you have factor 7, you get the 10. So from here, how is the common pathway? So 10 is along with the factor 5A will convert it into the thrombin, that is a factor 2. So how to remember is 5 into 2 is 10. This is also again in the descending order of the frequency, and here the multiplication will give you the 10. 5 twos is 10, and this thrombin will convert and form the fibrinogen. So factor 1 is fibrinogen. So 5 into 2 is 10, 5 into 2 into 1 is again, it is the 10. So remember that intrinsic pathway is in the descending order, extrinsic pathway is addition to the factor 3, and then the common pathway is multiplication, which leads to again the factor 10. Now, once the thrombin is formed, this thrombin will act on the soluble fibrinogen. Now, this fibrinogen, it forms the 4% of the plasma protein, but this form is soluble. So, it doesn't help in the formation of the clot. The thrombin acts on the soluble fibrinogen and it converts the soluble fibrinogen into insoluble polymerized fibrin. Okay, and then the thrombin also acts on factor 13, converting it into the active form. Now this activated factor 13A, it further acts on insoluble polymerized fibrin to form the cross-linking. So you have a cross-linking of the fibrin which forms a fibrin meshwork. This is how the thrombin acts, converting the fibrinogen to fibrin. So directly the thrombin will not convert it into the fibrin meshwork. Thrombin will convert the soluble fibrinogen into insoluble polymerized fibrin. Then later on we have the factor 13A, which plays an important role in forming the cross linkages between the fibrin, forming a fibrin meshwork. So I think now you remember this phosphatidylserine activating the coagulation cascade by activating the factor 12. So we have the factor 12A, then we have the extrinsic pathway which is activated, whereas the tissue factor will activate the intrinsic pathway. So both of them, the end product is the thrombin formation. Thrombin causes fibrin polymerization, and the fibrin polymerization later on it causes the stabilization of the platelet plugs. Now you have loosely attached platelets initially, but once you have a fibrin meshwork, the platelets will be present within the meshwork and there will be stabilization of the platelets. So that's what is called as stabilization of the platelet plug. Now, what are the other functions of the thrombin? So first of all, what we have seen is it causes the stabilization of the platelet plug. Another important function is it causes activation of the other coagulation factors like factor 9, 5 and factor 7. So whenever the coagulation factors are activated, you have the activation of the coagulation cascade. And another important function is it causes the activation and aggregation of the platelets. So through its ability to activate the PAR1 molecules. So you have the activation and aggregation of the platelets. Then another important effect is it has a pro-inflammatory effect. It has it goes and attaches to the endothelial cells, inflammatory cells, and other cell types, uh, and activates them. So these cells, they will be releasing the inflammatory mediators again. Then another important effect is it also produces the anticoagulant effect by combining with the thrombomodulin. If you remember what we have discussed, the thrombomodulin, which is expressed on the surface of the endothelial cells, it combines with the thrombin, and this complex will activate the protein C. So protein C combines with the protein S and it activates certain coagulation factors. So these are all the functions of the thrombin. Stabilization of the platelet plug, we have activation of the coagulation factors, activation of the platelets, activation of the inflammatory cells, and also it has an anticoagulant effect. Now, once the stabilization of the platelet plug occurs, in that you have only just the platelets. Initially, you had just the platelets which were present in the fibrin meshwork. So slowly what happens in this fibrin meshwork, the inflammatory cells, the leukocytes and the RBCs which are present in the circulation, they get trapped in the fibrin meshwork. So when these are trapped, the clot size is increased, and this platelet plug along with RBCs and the leukocytes, we call it as a blood clot. Now once the clot is formed, the function of the formation of the blood clot is to stop the leakage. So once the clot is formed, then there will be activation of the counter regulatory mechanism. Otherwise, what happens if there is no counter regulatory mechanism? Slowly the cells will be accumulating on the surface, and we have a very big clot which can even obstruct the flow of the blood in the vessel. So to prevent that, we have our own inner controlling mechanism. So once the clot is formed, we have the release of the tissue plasminogen activated from the adjacent endothelial cells. Okay, that was the step for the formation of the clot. Now the next step will be the clot retraction and the repair. So once the platelet plug is formed along with the leukocytes, the next step is there will be contraction of the platelets. Why this platelet contraction occurs is when the platelets contract, they pull the endothelial cells together. Now along with that, the platelets, when they contract, they also release certain growth factors. So we have the release of vascular endothelial growth factors which help in the regeneration of the lost endothelial cells. Along with that, we have certain platelet derived growth factors which causes the smooth muscle proliferation, and also this helps in the regeneration of the lost collagen. So in this way, the platelets, when they contract and when they release the growth factors, what they do is they bring together the endothelial cells. They help in the regeneration of the endothelial cells, and also the regeneration of the smooth muscle cells and the lost collagen. Now, once there is a formation of once there is a contraction and everything, then what happens? I told you that once the clot is formed, again there should be a control mechanism. So we have the release of tissue plasminogen activator. What does this tissue plasminogen activator do is it converts the plasminogen to plasmin. Now the plasmin, it has affinity towards the fibrinogen, and it causes the breakdown of this fibrin. Now the fibrin degradation products are called as the D-dimers. So once the plasmin is formed, it goes and attacks the fibrinogen or the fibrin molecules and causes them to degrade, releasing the D-dimers. Now if this continues, even the plasmin can cause damage. So in the circulation, once its function is over, the plasmin combines with alpha-2 plasmin inhibitor and it forms a complex. Now in this complex form, the activation of the plasmin will not be there, it will be inactivated. So once its function is over in the circulation, the plasmin will attach to the alpha-2 plasmin inhibitor, forming a complex where plasmin doesn't have any function or the plasmin is inactivated. Now that's why whenever there is a clot formation, the laboratory test what we do to find out whether the clot formation is there or not is the measurement of the D dimers, because whenever there is a clot formation, obviously there will be a fibrinolytic mechanism. So the fibrin degradation products should be present, and so D dimers should be present. If the D dimers level is raised, means in the blood there is a formation of the clot. Now, what are the factors which are limiting the coagulation? So already I told you, once the thrombin is formed and everything, it has an anticoagulant effect by combining with the thrombomodulin, which leads to activation of the protein C, and this protein C with protein S, it will inactivate the coagulation factors like factor 5 and factor 8. This is one mechanism. So in this way, once the clot is formed, it is again degraded, and then the thrombin which is formed, it will also be helpful in the anticoagulation effect only by combining with the thrombomodulin. Okay, this is the entire summary of the hemostasis. You see, these are the three steps in the primary hemostasis. That is, we have the vasospasm. Initial step, vasospasm. If the blood vessel is very small, even the spasm will reduce the blood loss. That's enough to reduce the blood loss. Later on, we have the attachment of these platelets to the von Willebrand's factor and the collagen, leading to the activation of the platelets. Then you have the activated platelets releasing ADPs, ADP, thromboxane and all that, which will cause the activation of the adjacent platelets and aggregation of the platelets by combining with the fibrinogen. Adjacent platelets, they adhere to each other by the fibrinogen molecule. Then we have a platelet plug formation. Later on, there will be activation of coagulation factors leading to the clot stabilization. Now this is clot stabilization. Later on, we have the formation of the blood clot, where you have the other formed elements also, like leukocytes and the RBCs. Now once you have the formation of the blood clot, there will be a control mechanism also. So this blood clot, once a blood clot is formed, the next step will be, there will be contraction of the platelet. So you have the clot retraction. Along with this, the clot retraction, there are also release of the growth factors by the platelets. Along with the platelet contraction, there will be release of the growth factors, which will help in the healing of the vessel wall. Now once this clot retraction and the healing process is over, the clot should resolve. So this occurs by the fibrinolysis, by the activation of the plasminogen. Tissue plasminogen activator is released by endothelial cells, which will be converting the plasminogen to plasmin, which causes the breakdown of the fibrin into the D dimers. And another mechanism is thrombin, which is formed. It will also combine with the thrombomodulin, which will activate the protein C, and this protein C will inactivate the coagulation factors, other coagulation factors. So the steps, if you remember, this is primary hemostasis, this is secondary hemostasis, and the tissue plasminogen activator, converting plasminogen to plasmin and causing breakdown, it comes under fibrinolysis. I hope till here the hemostasis is. Thank you, friends. I request you to give your opinion on the video, if any additions should be made or if any corrections should be made on this. Thank you.